So there was
points are but fine.
so there was gap of 30 minutes between the two sessions. The previous session ended at 11 a.m.
Okay, fair enough. You can start if you want, or you want to wait for.
Sir, if you permit, we'll
I'm
start our
sharing a
session.
screen by the time. I'll just check whether my screen is visible.
Okay. Okay.
But if my screen is visible,
No, sir. Yeah. Yes, sir. It is visible.
is my screen is visible?
Yes, sir.
Yes,
Drug
sir.
discovery facts
Please
and
start.
figures. Thank you, sir.
Sorry.
I again welcome all the participants on our lecture 2 of day 1 of AICT sponsored STTP on future trends in research methodology for drug discovery and development. In our lecture session 2, we have with us Professor P.R. Vavia. I would like to take this proud privilege in introducing the speaker to our participants who are faculty from various pharmacy colleges across the country. Professor P.R. Vavia is Dean Internal Quality Assurance and Professor in Pharmaceutics at Institute of Clinical Technology, Mumbai. Sir, on his B.Pharm, M.Pharm and PhD from Mumbai University in 1985, 87 and 91 respective years. Sir has more than 31 years of teaching experience to undergraduate, postgraduate and doctoral students. Sir has guided total 60 master students and 50 PhD students so far who are placed at key positions in leading pharmaceutical organizations. Considering his profound scientific knowledge and research expertise, Sir has been invited to various scientific conferences in India and abroad. His valuable inputs as a genuine academician, potential researcher, and versatile administrator were always acknowledged by various prestigious awards, to name a few. The Corporate of Science and Technology Award of Johnson & Johnson USA in 2001, UP Government National Award for an outstanding work done in the area of interaction with industries 2005, Best Teacher Award, University Institute of Chemical Technology in 2007, 2010, 2012, and 2014. Distinguished Teacher Award by Maharashtra Pharmacy Association in 2009, and Incentives to Meritorious Teachers, Dr. K.S. Ghada Reward in 2009. Vasvik Award in category of Biological Sciences and Technology for developing the novel drug delivery systems, synthesis and application of novel polymers and excipients and targeted drug delivery systems in cancer treatment, January 2015. With these words, I request Sir to please start his presentation. Sir, please.
Okay, good morning, of you. Am I audible clearly to all of you?
Yes, sir.
Very well.
Yes, sir, you are audible.
Very much. So you all can see my titles.
Yes, sir.
So at the outset, let me congratulate the Columbia Institute for organizing such a wonderful FDP and mainly on drug discovery. And I see many participants are the faculty members from various institutes, and I'm really thankful to Shukla sir for giving me this opportunity to speak. Unfortunately, I'm not from the area of drug discovery, but then I've been asked to maybe talk on drug delivery, and I said, okay, let me touch upon a topic on hot melt extrusion, which is a industrial technique nowadays in good demand. Unfortunately, it is not covered in our syllabus exhaustively. But then, yes, it is the technique which is widely used nowadays in industry, but very few industries are using. So the presentation will cover on drug discovery, the solubility issue, hot extrusion, SWOT analysis, formulation development consideration, technology aspects, and some case studies. And finally, I'll be summarizing my presentation. So since the theme of this conference is on the FDP is on drug discovery. So I thought initially I'll touch upon some slide on drug discovery. I'm sure these are preliminary slides, and you may be learning more than what I'm trying to show here. So please consider these as a preliminary slide, because I'm trying to link drug discovery with drug delivery system, and thereby these slides are there. So this is a very preliminary slide, and you have been learning in this FDP that in drug discovery, there are various stages like target selection, lead discovery, part of medicinal chemistry, in vitro, in vivo study, and finally clinical and therapeutics. So these are all part of say discovery, and there are various things one based on their expertise. So they do either a combinatorial chemistry or in silico screening, or trying to understand the mechanism of action, or trying to understand some animal model studies, so on and so forth. So, but what happened after the drug discovery? Okay? The facts and figures, which I said, though this is a slide which is till 2018, but more or less the picture is the same. So in this slide, right from 1993 to 2018, I have tried to touch upon which are the new molecular entities, or how many new molecular entities and how many biologically licensed applications are approved in these years. So if you try and understand the picture maybe after 2008, not that rosy. Okay. But you get in a year somewhere around 33 molecules as a new molecular entity which are approved. Maybe after 2017, 18, okay, there are more molecules which are approved. Okay, but then the actual fact is, if you see, again, this is a slide which is covering the diseases. So out of those 42 miles, many of them are concentrating on oncology or infectious disease or neurological diseases. Okay, so you see, out of 42, you may find around 36 molecules are in these three categories. But then if you see, out of these, many of these drugs, what is expected from drugs are they should have a good solubility. Okay, unfortunately, you have a problem with drug discovery is that after the drugs are discovered, not have a good physicochemical properties. So poor solubility, many of them. Okay. We look at maybe onco, we look at the selectivity, but then many of them are non-selective, toxic. We also look at these molecules need to be more stable, but many a time what happens is these molecules are unstable. And the patient considerations. So we see, so these are the actual, figures. What is we see in number of cases. I don't see all cases, but in number of cases. So what is expected, okay, and what is actual. So I'm going to touch upon these solubility issues and how hot melt extrusion, okay, can help in. So I'm going to touch upon this hot melt extrusion as a technique. So this is what is the expected discovery output. So maybe around 40% of the molecules, okay, you see they require to be modified with respect to physicochemical properties. Okay, so the existence of drugs and excipient in multiple physical forms, maybe either polymer, isomers, and so on and so forth, and maybe a solid form or a complex form. Okay. It's an opportunity, okay, to select the preferred form. So you have to come out with the best. Either you select the best drug, best polymer, and the best excipient to come out with the delivery systems. Okay, and try to make it as right as you can. Okay, so those 40% of those molecules, you require to change. You may get a 60% within the required physicochemical properties, but then those 40% which I'm trying to show here, okay, these need to be changed. Maybe, what I'm trying to maybe show you pictorially is well, it is not a circular, but then you try to make it circular by selecting the right type of a dosage drug and right type of an excipient. So the physical form of a drugs, if you see, either they are in the crystalline form or they are in the amorphous form. Okay, but generally, if you see in crystalline form, if you try and understand the physical pharmacy, the crystallinity part of it, the atoms in this solid are arranged in an orderly manner. So I try to show here that they are arranged in orderly manner, and the presence of long range translational and orientational order, okay, a long range orientation order, it gives them a crystalline structure. Whereas if you look at the amorphous form, okay, the amorphous form of a drug substance is characterized by disordered molecular arrangement. So they're not oriented in an orderly manner. Okay, so they're basically disordered molecular arrangement and lack of long range translational and orientational order. So higher drug solubility, maybe between 10 and 1600 fold, maybe then the crystalline form. So you can have an amorphous form, okay, but then there is a possibility that you may have the stability issue. Okay, so better mobility at a molecular level, which ultimately results in a better dissolution and the better bioavailability. So basically, what we are trying to maybe achieve is an amorphous form and also the stable form from a crystalline form, and try to improve upon the solubility. So you have the good alternative to a mechanical setup to enhance the solubility. So let us see a formulating in poorly soluble drug. So after the drug discovery, if you poorly soluble drug, what you generally do is develop a formulation with a stable amorphous form of a drug. Then it is not that easy, amorphous and the stability, it is not that easy. So it's a very challenging task. It's a very challenging task. So generally what people they do is how do they enhance the solubility? So either they change the particle size or they make some solid sharing, dispersion, micro emulsions, liposomes, complex, co-solvent, and maybe it's a factor system, we call it. These are approaches people have used, and one of the approach is using a solid dispersion technique. So in a solid dispersion, okay, what you try to do is, so among these people they select one of the technique which is a solid dispersion. So in the solid dispersion also, you have several techniques like spray drying, freeze drying, and maybe you have dehydration of a crystalline hydrates or precipitates, and end solvent addition, and maybe hot extrusion. So I'm going to touch upon one of the topic today, which can be used, okay, to enhance the solubility of a poorly soluble drug, okay, which is generally obtained after a drug discovery. So why hot extrusion? What you basically try and see. So in hot melt extrusion, generally this technique is a solvent-free manufacturing technique. Okay. Is my animation is going with my talk, or is there a lag?
No, sir, it is going along with the words. Okay,
Right. So you can also see my the video.
Yes, sir.
So the hot melt extrusion, if you see the advantages of hot melt extrusion, it's a solvent-free manufacturing. So many multinationals, nowadays coming out of a solvent, which is organic solvent basically. So this is one of the advantageous technique. There are very few processing steps, which I'm going to explain to you. It's simple and it can be made continuous. And also, believe me, the efficient process, okay, continuous and efficient process. And believe me, it is also an easily scalable to a production level. And one can have online process monitoring also. we call it as process analytical technique, or tools can be used. So the process can be monitored by using a PAT technique. So these are some beneficial points of hot melt extrusions, which one can look into it. But when you see hot melt extrusion, versus your conventional wet technique, okay, what basically in wet extrusion, if you see initially you do blending, and then you wet granulation maybe, and then you take out the granules, you try to spironize it, and maybe then you get those spheres, and then you dye them, okay, and then maybe you do a film coating and encapsulate or compress it. Okay. So maybe you put it in a cap or you compress it in a cap. So I'm trying to show you several steps which are part of wet extrusion process. But then in case of hot melt extrusions, there are fewer steps, as I said. One can do like milling, pelletization, spheronization, all in one. And one step you can do it, and then maybe you can, or you can fill it in a capsule. So you can see here there are less number of steps, and as I said before, this technique can be made stable very easily. So a kinetic of hot melt extrusion, if you see here, okay, what generally you get after the hot melt extrudes, okay, you get a release of a drag very easily, okay, maybe if it is hydrophilic drag, maybe a dissolution, and maybe an hydrophobic drags, okay, it may be a diffusion, okay, and so you have the liberation of a drug, and then it gets absorbed, it gets distributed, metabolized, and then the drug gets excreted. So then it follows your usual biopharmaceutics and pharmacokinetic. Okay, so it's like in any other solid dosage form. Okay, this can also be a technique can be used, okay, to develop a dosage form. So generally, a drug release, you see, this is a matrix what you get after a hot melt extrusion. So you have a polymer matrix in which the drug is embedded. So when it comes in contact with water, okay, so what it does basically, it tries to penetrate, and then when it penetrates, okay, it try and dissolve this drug, and the drug diffuses out. So it depends upon the polymer used. If it is a biodegradable polymer, it gets degraded, okay, very quickly, okay, based on the nature of the polymer. Now, if you try and see some of the commercial part of it or the intellectual part of it, the hot melt extrusion patents until 1983, you can see the major countries which are leading: Germany, okay, Japan, of course the USA is there. Now Germany and Japan, if you see these countries, and of course there are many European countries, and many of them are concentrating on excipients, okay, especially the polymer chemistry, and thereby they are trying to utilize their polymer in a hot melt extrusion. And thereby you can see Europe and Japan, they are in big number of patents. So, but then from 1983 onwards, if you try and see till 2011, the number of patents applications, in the area of hot melt extrusions have gone up. Okay, so they were below 5, okay, in 1983 onwards, and now if you see, okay, there is a good number of patents which are generated after 2011. There are many, okay, which have come up. So if you try and see here, the publications part also, many teachers may be interested, okay, to know how the publications in hot melt extrusion. So they're also increasing. There are a good number of publications you see in various countries, especially Germany and United Kingdom. If you see, okay, they are with near to US. Okay, but after US is more, and there are some other European countries. So after seeing some industrial part of this, you can try and understand the SWOT analysis of these hot melt extrusion. So the strength is the continuous process. It is okay, and it can be made reproducible, higher economic efficiency. So since there are less number of steps, the less energy may be involved, okay, and then thereby it is higher economic efficiency, we call it as. Okay, the wheel is not suitable for a thermolabile compound. High startup cost because the machine cost is a little higher, but it is one-time investment, believe me. And opportunity is this technique is environmentally friendly because there is no solvent Okay, so you're not talking about ozone layer depletions and all that. And wide range of dosage form can be manufactured. I come to the slide. It is not just a solid dosage form, but people are trying to make other dosage form also. And the threat is the knowledge and facilities are still a significant barrier, because the authorities like the FDA and all, okay, the regulatory agencies basically are still skeptical about the new technology, and there are a lot of cues about development, validation, quality control. Okay, so this may delay the approval process, but now there are many which have already come in the market as a hot melt product. So the regulatory authority very well works. So how do you do the development work? So the development involves the formulation part and the process part. Okay. So let us see key parameters. The drugs which have a solubility issues, the drug which require to have a proper melting temperature, physical thermal stability. These are the key parameters. While center molecules, the relevant characteristics of form components which goes with HMP is you require an API, you require a carrier which are polymer, and you require an additive. So API, when you say, is thermally stable. Okay, if it is insoluble drug, it is, it can be improved upon. One should look at the melting temperature, the miscibility of this API with the polymer, the particle shape and the size. What is the flow of these particles, moisture content, and the bulk density. And the carrier, which we call it as a generally a plastics material or a polymer. Okay, so thermoplastic behavior, the solubility again, there TG plays a very important role. Glass transition temperature, the degradation temperature. So they should not degrade. Many polymers degrade at the processing temperature. So to select the proper polymer, their dissolution properties, as I said before, preferably a biodegradable polymer, so that it gets degraded in a biological system. Since it is a polymer, it should have a low toxicity, and majority of the polymer, they vary in a batch-to-batch consistency. So they should have a proper reproducibility when we talk in terms of batch-to-batch consistency. And you require sometimes some additives like plasticizers you require. So the physical state of plasticizer, they should be compatible with API. Okay, their thermal stability, their melting temperature, their lubrication effect, and on and so forth. So these are some characteristics, which one has to look at as a formulation scientist when you work. So generally, what is the role of plasticizer in HME? So excipient that lower the glass transitions temperature of a polymer. So many a time plasticizer, when you add, it reduces further the glass transition temperature of a polymer. So the polymer melt a little lower temperature than what actually the temperature of a polymer is, and thereby your processing temperature goes down. Okay. So you can work at a lower temperature, lower energy. So allow processing at a lower temperature. Interaction with the polymer determines how much the TG is lowered. So how good is the plasticizer interacting with the polymer, and the effect of polymer mechanical properties. So it reduces the tensile strength, it improves the elongation temperature, elastic module. So all these mechanical properties of the polymer are modified by using certain plasticizer. And sometimes some drugs, okay, can also play like a plasticizer. Okay. So the drug also has some properties of a plasticizer. So the types of now solid dispersion. If you try and understand this slide, I'm sure you will understand these whole topic, very well. So this slide is very important. I tried to show some pictures here of how the crystalline form of the matter, or how an amorphous form and how molecularly dispersed a system look like. So basically, if you see, when the drug is crystalline, okay, and if the polymer, okay, is an amorphous, okay, and thermodynamic stability, if you have almost a stable, what basically you get is the glass suspension. Okay, you get a glass suspension. Okay, you don't get a glass solution. You need to have a glass solution. Like this. Now, when you have a drug which is amorphous, when you have the polymer which is amorphous, and if it is an unstable system, okay, it is a glass suspension. But when I say glass solution is, you should have a single TG, after you mix your drug and the polymer. So if it is molecularly dispersed, you get a stable system and which has a single TG. Now here, if you see the TG of a drug is different, the TG of a polymer is different. Okay, so here you get a 2 TG. Okay, but then if it is a molecularly dispersed system after the hot melt extrusion, that means the drug is completely dissolved in polymer solution, and thereby you get a glass solution. Okay, so this is the ideal system which one should get. Okay, if you want to achieve a good solubility, good solubility of a poorly soluble drug. Now, this is a slide which is an important slide which tells you about each system. When you try and mix drug and the polymer. Okay, when you say amorphous suspension means what, and when you say a solid glass solution means what. Okay, so I'm going to maybe first read out and then maybe explain you this graphics. So in amorphous solution, what you have is a molecularly dispersed. So your drug and polymer is molecularly dispersed. So there is an interactive force between A and B, which are weak. So the force between the drug and the polymers are weak. And thereby, the association between the drug and the polymer are weak. So majority of time get is the self-association. So the association is A and A, that is drug and between the polymer and polymer. So let me try and play this animation so that you will try and understand. Okay, you can see here what I'm trying to show here is there is an association, okay, between the drug. Okay, so that is the drug and drug. So you don't get a good interaction between drug and the polymer, and thereby you get an amorphous suspension. Whereas in case of in solid glass solution, okay, a molecularly dispersed drug and the polymer, which allows the interaction between drug and the polymer, which is a strong and which prevent the self-assembly of drug and drug interaction or the polymer and polymer interactions. Maybe by hydrogen bonding or maybe some weak forces which are. Let me play that animation and try and explain to you what you mean by solid glass solution. So in a solid glass solution, if you see, there is an interaction between the drug and the polymer, maybe with an hydrogen bonding or maybe some type of a weaker forces which are there. So the drug is maybe bound with the polymer. Drug is maybe, and thereby you get a molecularly dispersed system which is looking in a uniform matrix, which is nothing but a glass solution. So one has to get a very good glass solution system, then only you can get a better solubility of a poorly soluble drug. But then for that, you require to select a various carrier. These carriers are polymers. Okay, these are listed here. Now, believe me, what I'm speaking here is these are commercial polymers which are used or can be used in HME. Okay, can be used in HME. So I've shown here the temperature range of these polymers. We required cholecalcium VS solid. Okay, these are widely used polymer in HME. Okay, there are others which are also there. Because most of the tags, okay, the melting range which lies in this, okay, But then you have a wider choice. Okay, and there are many more. So as I said before, there are patents. There are literatures where these polymers are used. And if you look at many of these polymers are made by European countries. Okay, Okay, the manufacturers are from Europe. Okay, maybe Germany, you can see. Of course, the Japanese also involved, and US. So these are the wide range of temperature of a polymer. Like if you see Udrogit here, Udrogit can be used with a wide range of a drug. So if your drug is melting at a low temperature, you can use Udrogit. There are several grades of Udrogit can be used. Okay, you can use HPMC, acetate, and HPMC salt. HPMC derivatives. Okay, methocells, again an HPMC ethocells. Okay, Colidon, SR, this is another one, okay, which can be used. So you have a wide range of a polymer, but believe me, these are the polymer which are a commercial polymer. Okay, now I would like to maybe give some tips here. Okay, for an academic teachers. Okay, I am sure you people must be reading a lot of papers, a lot of publications and all. Okay, but then many a time it is up to you to understand when you read paper, which are the one the technology which are published in the paper, which are commercially viable, and which are the ones which are only for the publication purpose and has no commercial viability. I'll give you just one example, very interesting example. I'm sure since you people are in KDP and you will take this very positively. I was interviewing the faculty at a very high position in one of the college. So I went there as an expert. So I was interviewing one candidate having a 20 years of experience. Okay, and the candidate came for the professor post in pharmaceutics, and I was asked to ask some question to the candidate. And my first question to the candidate was, since you are into pharmaceutics, you may be teaching drug delivery system. So tell me whichever the topic which you like, and I would like to ask the questions. And the candidate says that the candidate is well versed with the transdermal drug delivery system. Believe me, I asked 10 questions, and all 10 questions were not having any satisfactory answers. Okay, I was asking about what which goes into a transdermal dosage form. Okay, as a commercial point of view, can you name some drugs which are commercial? Can you name some brand name of the product? Okay, can you name some products which consist of some known material, as an excipient? Okay, what is the role of backing membrane? What is the role of liner? What are these made up of? Absolutely no idea. Absolutely no idea. Absolutely no idea to the candidate. Okay, when you say you are expert in this particular topic and you don't have any commercial knowledge, but you don't have any knowledge about what is happening in the commercial market or what is happening in the industry, okay, in that particular area, the knowledge is of no use. Believe me. So maybe I'm sometime giving some messages which are not easily digestible to many faculty members. Okay, but then believe me, these are the factual situation today. Factual. I'm also an academic like you people. Okay, I have been teaching since last 30 years. Okay, but you need to change yourself. When you teach your student, you need to be more of an commercial, how it works commercially, how it works, rather than only talking of the industrial part of it. Let us see the technology aspects of ME. Okay, there are different extruders that are used. Okay, commercially, it's the single screw extruder. The name suggests it is only one screw. Okay, here you have a twin screw. So you can see here there are two screws which are there. Okay, and you have the multiple screw extruder. So there are several industries. They use this extruders. Can be food industry, can be a polymer industry, and of course the pharma industry nowadays start using the hot melt extruder. So a widely used is the twin screw extruder. Mixing, self-cleaning possibilities there, and the scale up is more predictable in case of an twin screw extruder. So you do R&D with the twin screw extruder, and then maybe you can scale it up by using a little higher version of a twin screw extruder. So you can use a small scale R&D, and you can then maybe scale it up by using a higher version of twin screw extruder. So these are some advantages. Okay, gentle mixing, excellent homogenization. There is a degassing and venting possibilities there, because if there is a gas generated, because when you are heating and mixing something, and if the gas is generated, you need to vent it out. Excellent process temperature control can be there. You can have a high steps and the low residence time, because it is moving through a particular zone. So if you have a residence time, if you keep it for a long time in that zone, I will come to that instrument later on. And there is a possibility your material may charge if you keep it for a long period of time. And there is an economic advantage because of continuous operations, less number of processing steps, one-step process. You can have a self-wiping, low material loss, less cleaning required, reliable process. You can have a PET process analytical tool and easily scalable to a production scale. So in which screw process, if you see here, self-cleaning is possible. Sorry. In single screw, this is a single screw extruder. After extrusion, you need to take out and then you can clean it. And in twin screw, they simultaneously get clean. I will show you there are different types of extruders. So I'm now coming to the machine part, the production part of it. So this is the process of a melt extrusion. So generally what you do is you add your polymer and the carrier. Okay, so your polymer and the carrier are put in the hopper. Okay, and then it runs through the hot melt extruder. Okay, so there is a mixing, melting. Okay, and the type of extruder which you require, extruders, you can pass it through maybe a milling, and then when you mill it, you get the extruders, which you can mix it maybe in a V blender with maybe other diluents and all, and then directly you can take it to a compressions. And if you require, a tablet, you can do it, or maybe you can straight away put it in a capsule. So believe me, these are less number of steps which are there and easily, workable steps. They are not that difficult steps. So there are screw elements. I'm talking more now of the machine part. Let me skip some of these slides. If you go into a detail of this, you will come to know about what are the different screws. This is what I want to touch upon. There are different configuration of this screw. If you see here, these are co-rotating twin screw. Okay, so they rotate in one direction, and thereby they are called as co-rotating screw. And there are co-rotating, but they're intermeshing. So the gap between these screw is less. And they are non-intermeshing, but where here they're intermeshing. So you can decrease the gap between these two screws. There are co-rotating non-intermeshing. Okay, so these are rotating in a different directions, and the other one which rotate again in the different direction, but then they're intermeshing. So the gap between them is less. So they have different advantages. When you start working with them, you will come to know what are the advantages. But then these are the configurations which are available. So these are the screw alignment which you can see. So there are different zones we call it as. Okay, we call it as a feeding zone. Okay, then the material is conveying. So you have a conveying zone. Okay, mixing. So in feeding and conveying, they get maybe further melted. And then there is a venting. Okay, so if there is a gas which is generated, it can be taken it out, and then finally you get extruders. So these are the different zones in HME, we call it as. Okay. Then coming to the screw geometry, this is more of an pharmaceutical engineering aspect. So what should be the screw geometry? So one should know about, there is a language used basically, what is the outer diameter, what is the inner diameter, what is the flip depth. Okay, so when you go and buy this machine, or when you work with these machines, people, they generally try to use the different screws. So you should know a little bit about the screw geometry. That is what I'm trying to emphasize here. Okay, and there's some steps. So when you feed something, what is the rate at which you are feeding? Okay, so that there is a unit of it, and what the power is required to force this. What is RPM? Okay, of the screw. Okay, what is the melting temperature, melting pressure which is there? Okay, finally, and what is the output which is coming? So your input and output, in terms of kg per hours, and there are some other parameters which you need to know. So it goes hand in hand, like what is your melting temperature. So depending upon the melting temperature you set, okay, if you set higher melting temperature, you may require a less force to extrude the material. If you have a lower melting temperature, you may have a higher melting pressure, and thereby your torque or a power which requires is more. Okay, so one should know when you work with HME, one should know some of these units. So now when you work with this twin screw extruder, one should know what are the independent variable and what are the dependent variable. What are the dependent variable? So the independent variable which goes like, a screw speed, feeding rate, the barrel temperature, okay, and then the screw design, barrel design, die design, and the processing parameters which are like melting temperature, residence time, and the quality control parameters like dispersions and the color. So finally, what you get. So these are some variables, which one can adjust when you work with HME. When you work with HME. And if you look at the scalable, as I said, it is a scalable because there are a number of machines which are available. You say that there is a Pharma Mini, which can be an R&D model. So if you work with Pharma Mini, okay, and it is a twin screw, okay, you can easily scale up. You can easily scale up, okay, from say a 10 to 50 grams to 0.5 grams and maybe to a 500 kg material. So you have a Pharma Mini which can work with, you can work at around 10 to 50 grams of batch size. And if you want to take a bigger batch, you can use a Pharma 11 by 16. And if you want to further have 500 kg batch, maybe you can take this. So from Pharma Mini to Pharma 2436, you can go very easily, believe me. Okay, because these are the machines which are designed so that you can easily scale up. Use easily scale up. So if you're working at an R&D, okay, you can easily scale up. Now, after telling you about some of the fundamentals, maybe I started with drug discovery, the challenges in drug discovery, and how a pharmaceutics person can improve upon the solubility by using a newer technique like HME. Okay, HME. So we saw now about what is HME, and what is this machine all about. Now, I will tell you quick about some of the applications. So you will get some knowledge about where one can use HME, So if you see, HME can be used in oral delivery system. In a transdermal patches also you can generate. So in oral you can make immediate release, you can make enteric, you can make sustained release, you can make control release. In transdermal you can make transdermal drug delivery, transmucosal, trans. And an implantable system you can make. Now, these are marketed formulas. Greaseophulvin, if you see here, greaseophulvin, like when we teach our pharmacy student, right, in the first year, maybe in a physical pharmacy, when we are talking of micromagnetics, So very good example which we try to give to our student is the greaseophulvin. When you reduce the particle size, there is an increase in the solubility. So a greaseophulvin, which was the product which was developed by using an HME. Okay, one of the very well known product today in the market is lopinavir ritonavir combination. These two drugs are highly insoluble drug in an HIV category. Okay, Caletra is the known drug. Okay, which is a tablet formulation, which is there in the market. Again, developed by HME. So like this, if you see here, there are several products which I have shown here, which are these are implants which are there. Ketoconazole is a delayed release tablet. You can see here. Again, the plain ritonavir. Okay. Then steroidal formulation, steroid, okay, using HME, azithromycin formulation, again using HME. This is a very interesting product, which is, we call it lacrisert. When you teach ophthalmic drug delivery system, lacrisert, it is nothing but a solid small rods put in the eyes, which are nothing but, there is no drug in it, and it is only hydroxypropyl cellulose. We call it as HPC, okay, ophthalmic inserts, which is used basically for dry eye syndrome. So you can see here, various category of products, okay, which are developed. Okay, you see an implant, you see a tablet formulation, a delayed release formulation, okay, and the ophthalmic products. And like this, you can make many more. Even you can make a transdermal patches, transdermal patches. Okay, so how do you characterize these once you get an HME extruded, what you do? So you do a thermal characterization like TGA, DSC, hot stage microscopy, microthermal analysis, atomic force microscopy, spectroscopy study you do, X-ray diffraction you do, then SME, TME you do, drug release in vitro dissolution study and do, and maybe you're required to do some biological properties. So these are in general the characterization of extruded, what you do. So after this, okay, let me quickly touch upon. I have now another 20 minutes. Okay, so quickly I will try and touch upon some literature on HME, where the HME is used. Okay, so increase bioavailability of poorly soluble drugs. So let me start with an example of ibuprofen with the melt granulations. So there is a carrier which is used with polymers, which has shown a very good increase in the solubility. If you see here, the melt granules, melt granules. This is the melt granules, which you get a uniform solubility. You can make a delayed release type of formulations also you can make. You can control the dissolution. Okay. Whereas if you can make a solid dispersion, you get maybe an immediate release. You may get immediate release. So you can control the dissolution profile maybe by using a polymers and ibuprofen. So there is a solubility enhancement. Okay. Then you have rofecoxib. Of course, the rofecoxib is that molecule now, but then it was a very good molecule. But people have worked with rofecoxib, and rofecoxib solid dispersion. Use polymers 1:5 ratio. So when you do it with hot melt extrusion, this is the solubility of rofecoxib. And you can see how good is the enhancement of the solubility of rofecoxib, which was achieved by using hot melt extrusion and the polymers. Then Toxicam and Eudragens. Okay. Believe me, these are again an example of commercial material, commercial material. So again, a piroxicam is a highly insoluble drug, and they have tried to achieve the solubility of piroxicam, higher dissolution profile they have achieved using a mixture of eudragens, and they have characterized by using X-ray diffraction studies. Celecoxib is another molecule which is of a ROF category COX-2 inhibitors. Okay, both these molecules, very good molecules when we talk in terms of COX-2, but they are not there because of the COX-2 limitations. But then you can see they had a very bad solubility, and they have achieved a good solubility. This is an example where you can increase the solubility by reducing the particle size also. Okay, so 0 to 120 micrometer, you can get a better solubility by reducing the particle size. Then you have a unique oral film, okay, of the paracetamol, okay, which can be put in the mouth, okay, and believe me, very efficient system. Okay, so the film can be prepared by using hot melt extrusion, and you can see here a therapeutic pictures of a salivary concentrations. You can see very good picture of paracetamol, good bioavailability. So not only for a solubility enhancement, but you can use for a test masking also hot melt extrusion. Again, they have used some anionic polymers, anionic API, and the ibuprofen. So ibuprofen, they have shown here a very good test masking ability of certain materials where ibuprofen can be masked completely. You can see here green is the no bitter test. You can have a no bitter test, so no unpleasant test. Okay, even if you have a granules of 2 grams in the mouth. Okay, 2 grams in the mouth. Okay, so they have used eudragit and ibuprofen. Eudragits and ibuprofen to test mask by using hot melt extrusion. There's another drug called verapamil. Cationic drugs, and they've used some eudragits, okay, which can interact with verapamil. And again, you can see here no bitter test, okay, with an 80 milligram of verapamil hydrochloride tablets, okay, for initial 20 seconds. Well, not only test masking, but you can use it for a controlled release formulations. The advantages of you have fewer steps at, you don't require a solvent, continuous operations, easily scalable, can be monitored online, and no compressibility requirement of the active. So if the active, even if it has a poor compression, you can still use it by using HME. So there is a drug called diltiazem hydrochloride. Again, then there are some commercial material which they have used like Puldon, Polydon they've used. You can see the picture with Polydon, a very nice release for 12 hours you can get with the diltiazem hydrochloride, a controlled release formulations. Okay. Theophylline, another molecule which is in the sustained release category, and people have used hydrogens. And you can see within hot melt extrusion versus the sterilization and extrusion. So you have a conventional extrusion and sterilization technique, and here is the technique of hot melt extrusion. Not only you get a spherical form, but you get a uniform picture, okay, of a hydrogen theophylline, and you get a better release out of this. So one can make a uniform system using HME, and they've shown a dissolution profile, a pH-dependent dissolution profile, which they've used with hydrogens. So you can see here a theophylline, a dissolution profile, becoming a very challenging molecule when you talk in terms of dissolution of a sustained release theophylline. It has a narrow therapeutic window, 10 to 20 microgram per ml. So you have to maintain the drug in that therapeutic window. If you go above therapeutic window, you get a toxic effect. If you come below therapeutic window, you don't get a theophylline effect. So theophylline is a very narrow therapeutic window. So very challenging molecule. You'll have to get the therapeutic levels in that range. Like this, there are several other examples. There is a carrier which is ethyl cellulose, guaifenesin, okay, ethyl cellulose. They have tried with. Okay. Then the processing temperature, how one can adjust the processing temperature and make robust formulation. So they've used a drug called ketoprofen, and they've studied what happened at 120 degree temperature of HME and 150 degree temperature of HME. How is the ketoprofen dissolution what you get, over a different temperature? So effect of temperature, using this HPMC polymer they've done. This is another drug which they have tried. So there are several molecules which people have done. This is guaifenesin. The previous one was acetaminophen, paracetamol it was. So you can see here again a commercial polymer like polycarbophil, PG3350, PK25, poloxamer 188. Again, then another example is nimodipine. This is again a calcium channel blocker, is a nasty molecule when we talk in terms of dissolution. Poorly soluble drug, nimodipine, nitrodipine, nifedipine, nisoldipine. All these are challenging molecules when we talk in terms of dissolution profile. They are highly insoluble. If you look at the monograph of nifedipine, they say in nifedipine, if you want to make, you'll have to add the carrier and make a solid dispersion. Okay. Without that, the nifedipine is not soluble. So it's like this, an example of PG with nimodipine. Okay. People have tried drug abuse prevention. There are a lot of patents by using HME, and they have tried to overcome the drug abuse. Okay. So HME is widely used in drug abuse. You can make a controlled release formulation, more controlled release. Okay. A fused matrix containing a drug and polymer, so that it is very difficult for the abused person, to take out this drug molecule from the matrix. It is not that easy. You cannot take out very quickly. So there are drugs, which are controlled medicine like fentanyl. Okay, fentanyl people, they might, extract the drug. Buprenorphine, again, a controlled medicine. So these are the drug abuse examples which you can make. Okay, and then there are some other advantages of ophthalmic of HME is aspirin granules. So you can improve upon the tensile strength of the granules by using HME. Okay, so they have tried measuring the strength of these granules. There are some machines which are there when you do a compressions. Then converting it to a direct compression material. So HME and a directly compressible material, how is the hardness which you get? So when you use a HME material and maybe a directly compressible material, what is that hardness which you achieve? So you can see the hardness. You can get a better hardness one can achieve. The tablet friability can also be improved. Okay, by you can have a less friability. Okay, as vis-a-vis a directly compressible material. Okay, you can try and play with the viscosity of the material. When you try different concentrations of plasticizer like triethyl citrate, here is an example of triethyl citrate, and what is the melt viscosity of the material, and thereby you can change the temperature which is required to take the material out. So you can play with the concentration of plasticizers and change the melting temperature of the material in HME. So one can play with this. There's another molecule called Ketoconazole, highly insoluble drug, antifungal drug. Okay, people have tried PVP solid dispersions. So they have tried pure API and solid dispersion, you can see by using HME. Okay, very good results which you get, completely amorphous. Okay, these are the XRD pictures. You can see here completely amorphous pictures of ketoconazole versus the crystalline plain API ketoconazole you can see here. So you can change completely from a crystalline to an amorphous form, and you can use an advanced analytical tool. As I said, process analytical technique one can use, like Raman spectroscopy you can use as a process analytical tool. So you can monitor the process. Especially there is one technique which is Raman spectroscopy, which is used to understand the film properties of the extruded films which comes out from HME. Okay, from HME. So I will show you that example here. So they have an online, you can see here, there is a Raman spectrophotometer, there is a probe which is connected, okay, to this film which comes out, okay, from a hot melt extruder. Okay, so you are putting material in the hopper, this is an extruder, and the mold which you have kept so that you don't get the extruded, but you get a film. And this film, the uniformity of this film, okay, the consistency of this film can be directly monitored using a Raman spectroscopy. So you can implement process analytical tool, that is a US FDA requirement if you want to monitor various processes. So the PET is an advanced analytical tool, can be used, and you can see if there is deviations. Okay, you can see the deviations which comes out with a larger noise. Basically, this is the output generally you see in near IR spectroscopy. So this is all about some examples which I wanted to give you. Okay, and I have another 6 minutes. Okay, so I will touch upon one example which a researcher has tried to improve the solubility and the dissolution of isradipine. Okay. So what is the work taken is the author has prepared and characterized. So how an R&D one can do, that is a case study which I wanted to tell. Okay. So what is isradipine? If you look at the isradipine as a molecule, okay, isradipine is a poorly soluble drug, antihypertensive drug. Okay, log P is 2.9. Okay, melting is 166 to 170. So you select a polymer in this particular range, okay, which also melt in this particular range. So they've tried a dissolution and isradipine physical mixtures. So isradipine and Colidon VS64, they tried different concentrations. So when you take a plain drug, when you take 1:1 or 1:5 and 1:10, what happens? So author have found when you use 1:10, just a physical mixture, there is an increase in the dissolution. And then what they did is they made an melt extrusion. So say from a 60% in 90 minutes, okay, author has achieved, in 10 minutes itself is almost 100% when you use 1:10 by a hot melt extrusion. So this was a physical mixture and this was after the hot melt extrusion. So there was a number of processing steps they have done and finally they have characterized, so this is how your Colidon physical mixtures and HME product look like. So you can see here the HME, okay, it is completely an amorphous extruded which you get. Okay, and believe me, they are also stable. Okay. And so you need to do a DSC analysis also and see whether they are glass solution. Okay. And SME studies, scanning electron microscopy study, a plain isradipine, a plain Colidon VS64, and the glass solutions. Okay, the closer view of glass solutions. Okay, how they look like. Okay, so you can see here there is a uniform dispersion. This is a closer view. Okay, So if there is a uniform dispersion you can see of isradipine and collidone VA64 in this in melt extrusions. And they've also tried another polymer which is called as Soluplus, okay, using HME. So Soluplus, you can see here isradipine Soluplus 1:10. You can see here the solubility you can achieve in 20 minutes almost 100%. Okay, isradipine Soluplus and isradipine Soluplus physical mixtures, how they behave. So HME definitely has an advantage, okay, for isradipine when you use Soluplus. So they have again done an XRD study to prove that it is an amorphous compound, completely amorphous. Isradipine solution, HME, Soluplus, it is completely an amorphous mixture after a glass HME. And the DSA studies, okay, which shows there is no endothermic pitch, which says their complete uniform distribution. And this is an scanning electron microscopy of the glass solution. Excellent. You can see here very uniform solution. And the stability part they have done. So you can see both they are amorphous, but the stability, okay, at 40-75, when you talk in terms of drug content, you talk of disintegration time, and you talk of dissolution T90 value, okay, less than 45 in all these cases. Okay, so it is remaining same. There is no change in this quality control parameters. So they remain the same. So that's about isradipine manufacturing process, which they have, selected, and they have tried these steps. Okay, extruded they've got, converted into a dosage form. Okay, these are how you get a long extruded if you don't break them. Okay, later on you need to crush them. Okay. So what you require is HME you require. Okay, that is an extruder. You require a dissolution. You require an UV spectroscopometer, and that's all. You can do easily this study. Okay, you can do this study. So my last slide is the reason we don't use newer technology is that we fear that FDA won't accept because of the agency is unfamiliar with them. But okay, believe me, this is a paragraph which is from a very well known person, okay, who is into we call it as the evaluation, the audit. When we do an audit, okay, of any process or any industry. So Emil is very, very well known person, analytical validations issue, where he has published and said that, okay, you work with the newer system, you make your FDA familiar. So majority of them, they don't work because they have a fear that the agency is unfamiliar with them. But the reason FDA is unfamiliar with newer technology is that the companies don't come out. Okay, so the responsibility is of a company, okay, to create that awareness. So unless and until you work with the new system, the regulatory authorities, okay, will not come to know and will not approve your product. Okay, so the more knowledge comes from the FDA, from the companies who work for the FDA. Okay, so with this, thank you very much for your patient listening. And if there are any questions, I'll be very happy to take them.
Any questions from participants? They can ask now. If participants are having any questions or queries, they can ask. Sir, I have one question with regard to this HME.
Yeah.
Sir, is there a lower processing temperature limit for this HME or this process? A lower processing temperature limit.
Sorry, can you repeat that question?
Sir, is there a lowering?
No, it all depends. No, see, it all depends upon what is the material which you have used. Okay, so If the low temperature material, if you want to take it out, as I said, thermolabile material, the material which are temperature sensitive, okay, which cannot be used. But then it all depends upon what is the carrier which you have selected and what is the TG of the carrier and what temperature you want to extrude the material. So the temperature at which you drug, and if it can come out, you select that particular temperature. So of course, there is a range which you can work, but then you can start maybe from a 40 degree, you can go till 180, 200 degree, depending upon what carrier which are available and what is the temperature stability of your API.
Thank you, sir. And sir, one more question that is
I
there
just
any...
wanted to know how many registered delegates are there for this FDP?
We have a number of 40 delegates which have registered themselves for this STTP.
And how many are attending today? Hello?
G, sir.
How many are attending?
Sir, we have at present 22 participants, 22 which have attended. And some participants they have joined, but somehow they left owing to the corrective issue or whatever may be the reason. But at present, we have 22 participants.
These are from the different institutions?
Yes, sir, they are from different institutes, from institutes.
Near Madhya Pradesh only?
Yes, sir, across India. Some of them are from Maharashtra, some of them are from MP, some are from Rajasthan, some are from South India.
Okay. So let me take your second question. Sorry, I had to interrupt you.
No, sir, that it is.
Yes.
The second question.
Somebody said
Yes, sir. The second question is: is there any correlation between this TG and TM values with HME?
You cannot immediately correlate unless you work with a particular machine. Okay. And when you work with the machine, you can start establishing the correlation. So it varies from a machine to machine. But of course, the material which you select and the melt temperature, okay, one has to record in a sense. So you can calculate what is called as melt flow index. Melt flow index. So the temperature at which it comes out, so you measure that, which is called as melt flow index. So there are small instruments which are there where you mix the material and pass it through a particular temperature, and you find out what is the flow required. So it gives you a calculation which tells you about what is the melt flow of that particular mix at that temperature.
And sir, one more question. This one is last from my side.
Yeah, please.
Sir, in one of your slides, you mentioned several polymers which are used in this etching. So, sir, I want to know that what characteristics must a polymer possess so that it could be used in this etching.
No, I've shown one slide where the API characteristic, the carrier characteristics, which I said.
Yes.
Okay. Do you want me to go back to that slide? I have said that. And the third one is the additive. So the carrier characteristics, which I said, thermal stability, all that which I said. So it should have a proper TG. Okay. So then only one can use it. Okay, it should be inert.
Thank you, sir. But as this topic is new for me a bit, that's why I have posed some of the questions. I was not aware not aware with respect to this technology.
Thereby, I thought that I will select the topic today. Maybe I've been speaking on HME at various forums. So this part of India I have not spoken. So I said, let me speak.
Any more questions from participants? I think, sir, there are no questions now from participants. So on behalf of Columbia Institute of Pharmacy, I thank you very much that you spared your valuable time and gave such nice presentation on the topic. Participants must have benefited themselves by hearing you. Thank you, sir.
I'm sure you will. Okay. I have one question. If you have a recording of this, you can please send it to me.
Yes, sir. We are recording the meeting. I'll send it to you.
Okay. Okay. I will also get this recording of the attendance also. Correct. Fine. So thank you very much once again for giving me this opportunity. Okay. Bye-bye.
Thank you. Thank you very much. After this lecture series, we meet again at 1:30 p.m. with our third lecture of day 1 of AICTE sponsored STTP on future trends in research methodology for drug discovery and development. I request all the participants to join again 5 minutes before the time schedule for the lecture so that we can start with our lecture 3 of day 1 AICTE sponsored STTP. Thank you.